Friends, welcome back. So, the next class of the flavonoids is chalcones and neurons. Now, chalcones are nothing but these are the open chain flavonoids. It means that they do not have any heterocyclic ring present in their structure, or I will say they do not have a pyrene ring in their structure. So, as you can see the basic nucleus on the slide, though they still have the basic carbon skeleton of C6, C3, C6 structure, but the pyrene ring is absent and they have an open 3 carbon chain which attach the 2 phenyl rings in the structure. And since the double bond present in the 3 carbon chain, it is having a configuration of alpha beta unsaturated carbonyl system in the chalcones. Now, since they lack the heterocyclic ring in their structure, but these chalcone compounds can be easily converted into the flavonoids under the acidic condition. And the common example of chalcone type of flavonoid is Liquidity Genin. It's an A-glycone form, and Liquidity Genin is largely found in Glycyrrhiza glabra. And Liquidity Genin is a simple type of chalcone, and if the chalcone is substituted with the prenyl chains or the other side chains, as we can see in Bava chalcone, which is largely found in Sorelia corallifolia, which is used for psoriasis, and such structures are the complex chalcones. Now, chalcones are also known as precursors for different types of flavonoids, which we will discuss in the biosynthetic pathway of the flavonoids. Now, the neurons are the flavonoids which are having 2 benzylidene cumaronone structure. We can see the basic nucleus of neuron. Now, neuron have a heterocyclic ring with a 5 atom heterocyclic ring, where one is carbon and the other 2 are one is oxygen and the other 2 are carbon. And the third carbon of the C3 skeleton is attached on the outside of this heterocyclic ring. So, the major difference between the neuron and the basic flavonoidal structure is that here we have a 5-membered heterocyclic ring, whereas in the general flavonoidal structure we are having a 6-membered heterocyclic ring. And the most common example of neuron is lyco-agro-neuron, which is again found in Glycyrrhiza glabra. And when these neurons are treated with alkali, the color of neurons, which is normally golden yellow or yellowish color in the plants, they changes to red in color. Now, the next class of flavonoids is pterocarpans, and this is the last class of flavonoids. And pterocarpans have altogether different type of structure, and they have a tetracyclic ring structure where they have 2 heterocyclic rings. One is 6-membered and other is 5-membered. And pterocarpan structure is derived from the basic isoflavone skeleton by the ether linkage between 4 and the second 2' position of the basic isoflavone nucleus. Now, majority of the natural pterocarpans are obtained as phytoalexins. It means whenever there is an infection in the plant or a fungal or a bacterial exposure to the plant, then plants start producing such compounds to protect itself against those microbial attack. And pterocarpans also has a very significant antioxidant property, which also protect the plant against various kind of physiological stress condition as well as the microbial attack against different pests. Now, pterocarpans are categorized into simple pterocarpans or 6-alpha hydroxy pterocarpan flavonoids and pterocarpine flavonoids. And the common example of pterocarpans is picetin, which is a 6-alpha hydroxy pterocarpan, and this type of pterocarpan is largely found in the pea plant. So, friends, this was about the different classes of flavonoids. Now we move on to the biosynthetic pathway of the flavonoids, that how these flavonoids are synthesized within the plants. Now, flavonoids are synthesized with the help of an amino acid known as phenylalanine, and phenylalanine is an amino acid which is produced after shikimic acid pathway. And the first step in the biosynthesis of flavonoid is that this phenylalanine is acted upon by phenylalanine ammonia ligase enzyme, and as the name indicate, ammonia ligase, it means this enzyme is responsible for the removal of the ammonia group from the amino acid, that is phenylalanine. And here you can see in the slide, the enzyme removes the ammonia group and it gives rise to trans-cinnamate or the trans-cinnamic acid. In the second step, this trans-cinnamic acid or cinnamate, it gets converted into trans-4-coumarate or paracumaric acid by the action of cinnamate 4-hydroxylase. Now, as the name of the enzyme indicates, cinnamate 4-hydroxylate, it means it will add a hydroxyl group at the 4th position of the phenyl ring, as you can see in the slide. Now, this trans-4-coumarate further gets converted into 4-hydroxycinnamic acid CoA. This happens by the action of the 4-coumarate CoA ligase enzyme, and this reaction occurs with the help of an ATP molecule. Now, this conversion of trans-4-coumarate into 4-hydroxycinnamic acid CoA, it occurs in 2 intermediate steps. In the first step, the parent molecule, that is the trans-4-coumarate, reacts with ATP to form an adenyl intermediate, and in the second step, this adenyl intermediate cleaves and AMP is released, and the remaining intermediate, it gets combined with the sulfhydryl group of coenzyme A to form the 4-coumaryl CoA or the paracumaric CoA. Now, in the fourth step, the 4-coumaryl CoA that was formed during with the help of ATP, this molecule, that is 4-coumaryl CoA, this acts as the major branching point in the flavonoidal biosynthetic pathway, and we can say this compound also act as a precursor for different flavonoids. Now, on the one side, it reacts with 3 molecules of melanoyl CoA by the action of stilbene synthase, and it gives rise to stilbenes. Now, stilbenes are important bioactive molecules, and the most common example of a stilbene compound is resveratrol, and resveratrol, we all know, is found in the skin of grapes, and by its potent antioxidant as well as its action on other molecular markers in the body, resveratrol has shown numerous actions specifically on cardiovascular system, protective action on cardiovascular system. This 4-coumaryl CoA again combines with 3 melanoyl CoA molecules, and this reaction is catalyzed by Chalcones synthase as well as polyketide reductase enzyme, and these enzymes give rise to 6-deoxychalcone molecules. Now, Chalcones synthase and polyketide reductase, so Chalcones synthase undergo or give rise to series of hydroxylation or condensation reactions, and it give rise to a polyketide intermediate, and this intermediate is acted upon by polyketide reductase, which leads to the formation of ring A in the flavonoidal nucleus, as you can see in the structure of 6-deoxychalcones. On the third side, this 4-coumaryl CoA, as I told you, this is the main branching point. On one side it is giving stilbenes, on the other side it is giving 6-deoxychalcone molecules, and on the third side it again reacts with 3 molecules of melanoyl CoA, and it gives rise to naringin chalcone. Now, this reaction is catalyzed by Chalcone synthase. Now, again, Chalcone synthase cause a series of condensation and hydroxylation reactions which give rise to this naringin chalcone molecule. Now, up till now, this is a brief summary what we have discussed in the biosynthesis of flavonoids, starting with phenylalanine acted upon by ammonia lyase enzyme. giving rise to cinnamic acid, 4-fumarate acted upon by then 4-hydroxylase enzyme, then 4-fumaryl-CoA is formed. 4-fumaryl-CoA is the main branching point. On the one side it is giving rise to stilbenes. On the second side, with the help of chalcone synthase and polyketide reductase, it gives rise to 6-deoxychalcones. And on the third side, again with the 3-malonyl-CoA molecules, it react with chalcone synthase to give rise to naringin chalcone. Now from naringenin chalcone, various other classes of flavonoids are synthesized. So in step 5, naringenin chalcone again acts as a branching point, and by the action of A-urosidine synthase enzyme, it gives rise to A-urons. Whereas it is also acted upon by another enzyme which is known as chalcone isomerase, which causes the cyclization of the open ring in the naringenin chalcone, and it gives rise to a type of flavonoids which are known as flavanones. And since we are starting with naringenin chalcone, the flavanone which is obtained is naringenin, as you can clearly see in the structure. Now chalcone isomerase is enzyme which is very stereospecific, right? So in the next step, that is the sixth step of biosynthesis of flavonoids, this flavanone molecule again it is having two branches. The naringenin flavanone molecule is acted upon by flavone synthase 1 and 2 enzyme. Now these flavone synthase 1 and 2 are responsible for the introduction of unsaturation in the ring C of the flavonoid nucleus. As you can see in the slide, the naringenin is acted upon FNS1 and FNS2, and there is no double bond in naringenin, which is a flavone, and after action upon by flavone synthase, there is an introduction of double bond in the ring C. On the other side of the pathway, naringenin is also acted upon by flavone 3-beta hydroxylase, that is F3H enzyme, and it gets converted into dihydroflavonol molecule, and here in this we have taken example of dihydrocamphorol. Now again this flavone 3-beta hydroxylase, it is a stereospecific enzyme and is responsible for the introduction of a hydroxyl group in the flavanone molecule, which is naringenin. So you can see in the slide, naringenin, by the action of F3H enzyme, there is an addition of a hydroxyl group at third position. As I told you, it's a stereospecific 3-beta position, and there is an addition of the hydroxyl group. Now in the seventh step of the biosynthesis, this dihydroflavonol compound, which was synthesized, that is dihydrocamphorol molecule, it is acted upon by flavonol synthase on one side. So earlier flavones were formed from flavanone, and now from the dihydroflavonol, that is flavanonol, we are synthesizing flavonol. So again it is acted upon by flavonol synthase, that is this enzyme will help in the synthesis of flavonol and introduction of a unsaturation in the ring C between second and third carbon. On the other side, this dihydroflavonol molecule is acted upon by dihydroflavonol 4 reductase enzyme, and it leads to the synthesis of leucoanthocyanidins. Now DFR is the enzyme which is responsible for the introduction of hydroxyl group at position 4 in the ring C. You can clearly see in the slide, the dihydroflavonol molecule is having a ketonic group at position 4 in the ring C, and by the action of DFR, it gets a hydroxyl group at 4 position in place of ketonic group, and we get a leucoanthocyanidin structure. Now leucoanthocyanidin is again acted upon by anthocyanidin synthase, which leads to the unsaturation at carbon 2, 3 and 4 in the structure, and it gives rise to anthocyanidin molecule, which is pelargonidin. Now leucoanthocyanidin, by action of anthocyanidin synthase, it also gets converted into 2-flavine 3,4 diol molecules. So as you can see, the anthocyanidin synthase, basically it introduced the unsaturation between the second and third carbon of the benzopyran nucleus. Now leucoanthocyanidin are also acted upon by the reductase enzyme, which is known as leucoanthocyanidin reductase, to give rise to flavonthreol molecules, and these flavonthreol molecules, they undergo oligomerization and polymerization and give rise to proanthocyanidins, which are also known as condensed tannins. Now anthocyanidin, which was produced during the 8th step, is also acted upon by anthocyanidin reductase, and by this action it again gets converted into flavonthreols, which further polymerize or oligomerize to give rise to proanthocyanidins or the condensed tannins. Now this is the complete overview of the flavonoidal biosynthesis, where you can see how the 4-coumaryl CoA, which was obtained from phenylalanine, and it gave rise to different types of flavonoids, and first it gave rise to chalcones, then flavonones give rise to flavones, then flavonones also give rise to flavanonols, which further give rise to flavonols, then proanthocyanidins, leucoanthocyanidins, and anthocyanidin. So this is the overall biosynthetic pathway of the flavonoids. Now we move on to the extraction of flavonoids, and since flavonoids are largely these compounds occur in the glycosidic form, they have a tendency to get degraded by the enzyme if the plant material is not dried properly. So the prime requisite, if you want to extract the flavonoids from a particular plant, so the prime requisite is that you should immediately dry the plant material after collection, so that all the moisture and the enzymes which can cause hydrolysis of those glycosides get deactivated, and you can then extract the flavonoids. Now the selection of the solvent to extract the flavonoids depends upon the type of flavonoid you want to extract. So if you want to extract a polar type of flavonoid, then you have to use a polar solvent. If you want to isolate mostly A glycons and less polar flavonoids, then we normally use a medium polar solvent such as chloroform, ethyl acetate, and so on and so forth. Now, as we were discussing that the less polar flavonoids like isoflavones, flavanones, or methylated flavones, and some of the flavonols, they can be extracted with the help of chloroform, dichloromethane, diethyl ether, or ethyl acetate. Whereas flavonoid glycosides, which are more polar by virtue of the large number of hydroxyl groups, as well as the more polar A glycons, mainly hydroxylated flavanols or biflavonoids, A-urons, or chalcones, they can be extracted with the help of acetone, alcohols, or alcohol water mixture in different percentages. Now some of the flavanone molecules, along with some chalcone glycosides, they are very difficult to get extracted with methanol or ethanol or alcohol water mixtures, and for them we normally play with the pH of the solvent which is being used for the extraction. And for an example, flavone solubility is normally affected by the pH and pH of the solvent, and when we have to adjust the pH, we normally use a water based extracting solvents. Likewise, flavonthreols, catechin or their oligomeric or polymeric forms, which are proanthocyanidins or the condensed tannins, they can be extracted directly with water, because when these compounds get oligomerized or polymerized, there are large number of hydroxyl groups present on their structure, which make them highly polar and water soluble. And the anthocyanin type of flavonoids, they are normally extracted with acidified methanol, and the different acids which can be used is up to 7% of acetic acid or trifluoroacetic acid, which is used up to 3%. Now this is the overall chart for the extraction of the flavonoids, and the first step is we take the dried plant material, and we can extract this plant material with either ether or hexane in order to remove the interfering compounds like pigments or carotenoids or lipids or fatty substances. So hexane or ether treatment helps in removing those interfering substances. And then we get a petroleum ether or hexane extract, which is normally discarded, and then the mark is again treated with a suitable solvent depending upon the type of flavonoid you want to extract. So if you want to have less polar flavonoids like isoflavones or flavanones or methylated flavanones, then we go for lesser polar solvents like chloroform or dichloromethane or ethyl acetate. And if you want to extract highly polar flavonoids or the glycosidic flavonoids, then we go for more polar solvents such as methanol, ethanol, or their aqueous mixtures. Now, once these extracts are produced, these are again treated with lead acetate. Now, this treatment with lead acetate helps in precipitation of the tannins, which normally also get extracted during the treatment with different solvents. Now, once these tannins are precipitated, the resultant mixture can be filtered to filter out the precipitate tannins, and the filtrate will contain the respective type of the flavonoids in their respective extract. Now, at this stage we get a crude extract which is rich in flavonoids, and from these crude extracts, if you want to further isolate or purify the individual flavonoids, then you can put on these extracts onto various chromatographic techniques, and the most commonly used techniques are the column chromatography or the centrifugally assisted preparative chromatography or the preparative HPLC or flash chromatography. So by using all these chromatographic techniques, you can isolate the individual and the pure form of individual flavonoids. Now, apart from this flow chart, the extraction, the plant extraction can be done with the help of different extraction methods such as maceration, soxalate extraction, pressurized solvent extraction, which is also known as accelerated solvent extraction, then ultrasound assisted extraction or supercritical fluid extraction. Now, apart from maceration and soxalation, all other techniques like pressurized solvent extraction, ultrasound assisted extraction or supercritical fluid extraction, these techniques have shown a very enhanced extraction efficiencies. Vis-a-vis, they also preserve the actual nature of the structure of the flavonoid without causing any kind of degradation to the original structure of the flavonoid. So this is how basically you can extract the flavonoids from the plant matrix. Next, we have the chemical test by which you can identify or determine the presence or absence of the flavonoid in the plant matrix, and the most commonly used chemical test is Shinoda's test. And in this test, the alcoholic extract of the plant is mixed with magnesium turning or magnesium ribbon, and to this mixture concentrated hydrochloric acid is added, and if flavonoids are present, the color of the solution will change, will have a color range of orange to red, and if orange to red color is obtained, that will indicate the presence of flavonoids. Likewise, we can also detect the presence or absence of flavonoids with the help of zinc test, and in place of magnesium in this test we add zinc turnings or zinc ribbon, and concentrated HCl is added. We obtain a deep red color in the end, which indicates the presence of dihydroflavonoids, or we can say flavon type of flavonoids. Third test is the ammonia test, where we dip a strip of filter paper in the alcoholic extract of the plant, and then we expose that strip of filter paper to the ammonia vapors. And when this strip is exposed towards the ammonia vapors, a yellow color of the paper strip is produced, which indicates the presence of flavonoids. And the third and the fourth test is of vanillin HCl test, or the hydrochloric acid test, where the alcoholic extract of the plant is treated with the vanillin hydrochloride solution, and a pink color is obtained in the resultant solution, which will indicate the presence of the flavonoid in the plant matrix or the plant sample which you have got. Now last we come on to the uses of flavonoids in the plants. Now as we already discussed that flavonoids are largely obtained as phytoalexins within the plant, specifically some of the classes of the flavonoids which we discussed today. So in plants, the major function of flavonoids is to protect the plant from various microbial attacks, mainly by their antimicrobial, antifungal or antibacterial action, and that is why some of the members of the flavonoids, they are considered as phytoalexins. Besides this antimicrobial action, flavonoids are also having a strong antioxidant action, which also helps in protecting the plant from the attack of different types of pests. Now we move on to the uses of flavonoids in the biological systems of man. And as I told you that these are the molecules which are having large number of hydroxyl group on their structure, and by virtue of these hydroxyl groups, these flavonoids possess a very potent antioxidant action. And this antioxidant action basically helps in quenching the reactive oxygen species. And we all know that these reactive oxygen species are very harmful to the biomembranes that are present in our body, and once these membranes are damaged, the whole of the system collapse. So flavonoids basically quench these reactive oxygen species. They prevent these reactive oxygen species from harming the biomembranes, and by virtue of this antioxidant action, flavonoids have exhibited numerous biological activities, and they have shown protection against neurodegenerative disorders like Alzheimer's disease, Parkinson's diseases or atherosclerosis. Then they have also shown potent action against different types of cancers. They have shown action in various cardiovascular disorders. They have exhibited a very potent anti-inflammatory action by mitigating the effect of pro-inflammatory mediators or markers present in the body. They have also shown protective action in various CNS disorders like anxiety, epilepsy, and gastroprotective, hepatoprotective and antimicrobial action they have also exhibited. So friend, this was all about the flavonoids, and we have discussed right from their definition to their classification to their properties to their biosynthesis, chemical test, and finally we discussed the uses of these flavonoids. Thank you.